We have seen up to this is that in case of transcellular mechanism, we have gone through passive diffusion, ion pair transport and the pore transport. And then our next mechanism is the facilitated diffusion. I have written here facilitated diffusion and active transport. Why? Because this both mechanism is based on the carrier. And this carrier, what are the properties of this carrier? These all are the listed properties of this carrier. And in case of carrier, first, they all are the proteins, so it can interact. Now, this indicates that these proteins are bidirectional. There is nothing direction says that. So, if this one is the membrane and only this protein can transport this side only, it can transport this side also. So, it is the bidirectional, no unidirectional protein is there. Now, next is it is based on the lock and key. That we know that particular lock is fixed with the particular key. So lock and key model is there, but duplication is also there. So sometimes false substrate can also bind. Generally, this all are the mechanisms. When protein is there, we go for the general structure of protein. That is R, C, this acid, this ammonia, and H. This is general structure of protein. In case of protein, it is made up of amino acid. It is general structure of amino acid. So here, some CONH bonds are there, and it will take place here also. So what happens here? That when such reactions takes place, this open chain interact with the particular type of substance. Suppose this chain, my protein is made for the finding the molecule which is having the simple, we consider that the methoxy group. Suppose if methoxy group is present in the drug, my molecule can bind. Okay. So this is the ligand for the particular protein. So now suppose R1, this is my drug, and some other substance which is also having this methoxy group. This R2. Okay. So this both can find way to bind this particular carrier. So generally this recognition site is there. That will find the methoxy group, it can bind. The other part, except for steric hindrances and other factors, it will not affect. If ligand is there, it can easily bind. So false substrate may enter in the body by means of this mechanism. Now, once you can see that molecule 1 and 2 types of molecules there, so there must be the competition between them for binding. So it is the competitive mechanism. Now, as the carrier is there, suppose I am having the 100 carriers. 100 drug carriers are there, and I am giving the drug which is having suppose 600 molecules of particular drug. These all are the hypothetical things, that suppose 100 carriers are there and 600 molecules are there. So what is going to happen? If I plot the graph of binding against the action, so what happens? At initial stage, the transport will be of first order, but once 100 molecules will bind, I can write this as 100 plus 500. So this 100 will bind to this, then after this 500 molecules are there. So what happens to this molecule? So now it will show no further increase in the rate of transport. Now this will be steady. As this one of this molecule will release, it will again 99 and one will transfer from